Hello, I am Tyler Roach, and I'll be presenting the original method of dideoxy DNA sequencing. So, what is dideoxy DNA sequencing? Well, let's start with the basics. So first, dideoxy DNA sequencing was created by Frederick Sanger and his colleagues in 1977. This particular method of sequencing will be later known as the Sanger sequencing method. In this method, the DNA is sequenced based on selecting and incorporating chain-terminating dideoxynucleotides via DNA polymerase during in vitro DNA replication. In vitro basically means that we're going to be performing this DNA replication in test tube experiments or experiments that use flasks, petri dishes, and micro pipetter plates. Now, to understand Sanger's sequencing method, we need to know the difference between a regular nucleotide and a dideoxynucleotide. A nucleotide is a sugar attached to a phosphate attached to a nitrogenous base, which is either adenine, thymine, cytosine, or guanine. And what makes a regular nucleotide is that it's attached to two hydroxyl groups on the 3' and the 2' ends. A deoxynucleotide is when you do not have the hydroxyl group on the 2' end. Deoxynucleotides are also known as DNTPs. When DNA polymerization occurs, a reaction between the hydroxyl group on the 3' end and the phosphate on the new DNTP produces a reaction. This reaction allows the new DNTP to be added to the nucleotide. Remember that a regular nucleotide has hydroxyl groups attached to the 3' end and the 2' end of the nucleotide. A dideoxynucleotide is when there is no hydroxyl group on the 2' end, and a dideoxynucleotide is when you have no hydroxyl groups on both the 3' end and the 2' end. While DNA polymerization is occurring and a reaction happens between the 3' end of the old DNTP, or the old part of the nucleotide chain reacts to this dideoxynucleotide, phosphate, you can see that the dideoxynucleotide is added to the nucleotide chain, but because there is no hydroxyl groups present on either the 3' or the 2' end, no further nucleotides, no further DNTPs can be added to the nucleotide chain. This stops DNA polymerase reactions and also results in chain termination of the nucleotide. Dideoxynucleotides are also known as DDNTPs. So how does Sanger's sequencing method work? Well, Sanger and his colleagues had 4 different test tubes. In all of the 4 test tubes, you had a DNA template, a primer, DNTPs, and DNA polymerase as the 4 main components. The difference between the 4 test tubes would be the different DDNTPs that would be in each of the 4 different test tubes. Test tube 1 would have DDATP. Test tube 2 would have DDGTP. Test tube 3 would have DDCTP. And test tube 4 would have DDTTP. This is what made the 4 test tubes different. In the 4 test tubes, the DD and TPs were also radio-labeled. This radio labeling would help us to later see the sequence of the DNA that was fragmented. Using test tube 2 as an example, you can see inside test tube 2, you have your DNA template and your primer. Also in your test tubes, you have your high concentration of different DNTPs and your low concentration of your DDNTP. In this case, in test tube 2, you have DDGTP. DNA polymerization occurs, and you have the adding of different nucleotides to the DNA template. But remember, when you have a dideoxynucleotide added to the nucleotide chain, and remember that the DDNTP is also radio-labeled, this addition of the DDNTP will stop DNA polymerization, and no further addition of nucleotides will occur. This results in chain termination. Because the addition of DDNTP and chain termination is very random, inside of your individual test tubes, you can have a ton of different fragmented DNA sequences, and these DNA sequences can also be different lengths. However, if you know, say, the length of the DNA sequence, you will know where your nucleotide, the nucleotide location is. So again, in this case, we have DD GTP, and this DNA sequence is 1, 2, 3, 4, 5 bases long. And so, since this is 5 bases long, we know that guanine is located on the 5th base. This same process is repeated for all the 4 test tubes and will be later used in electrophoresis. After fragmenting the DNA in your 4 test tubes, you can heat denurture the DNA fragments. And once these fragments are heat denatured, you can add the fragments to an X-ray film or polyacrylamide gel. In our example, we're going to be using the polyacrylamide gel. And the polyacrylamide gel is used to see partial DNA fragments from each test tube reaction. In the polyacrylamide gel, the contents of each test tube will be placed in one of the 4 wells. Then, during electrophoresis on the polyacrylamide gel, we'll separate the DNA fragments of each reaction. This separation of the fragments is based on the size of the fragments from each test tube. Larger fragments will be placed near the top, while the smaller fragments will be placed more near the bottom of the gel. We can then use the results of electrophoresis to read or see the DNA sequence. Remember that all of our 4 test tubes were radio-labeled. Remember that test tube number 1 was DD-ATP, test tube 2 was DD-GTP, test tube 3 was DD-CTP, and test tube 4 was DD-TTP. To determine the DNA sequence, we read from bottom to top, and we'll start with this one since it's the lowest. We will start from the shortest and then go to the second shortest, and then to the third shortest, the fourth shortest, the fifth shortest, and so on. We can now read our DNA sequence as G, T, C, A, T, G, C, A, G, A. Again, remember that I read the DNA sequence from bottom to top. And now we know the sequence of the newly synthesized strand. You can also use the synthesized DNA sequence strand to figure out what the sequence of our template strand, the original template strand, is. Our original DNA template strand should be complementary to our synthesized DNA strand. So our complementary original DNA template strand should be T, C, T, G, C, A, T, G, A, C. And this completes the Sanger sequencing method, or the original method of dideoxy DNA sequencing.